All right. Well, I'd like to, first of all, introduce myself and thank everyone for being here. My name is Kurt Farrell. I am an assistant professor at Mount Sinai, also working in the digital pathology space. So I'm going to take over some of the emceeing of this particular webinar from Dr. Duggar and Dr. Murray, and also Dr. Flanagan, who have put on this for many months leading up to this point, and I really thank them for getting this initiative started. We have a really interesting talk today by Dr. Kapasi at Rush. Just a little background on her. She is a research neuropathologist. She's obviously at the Rush ADRC, assistant professor. She has a PhD in neuroscience from the University of Sheffield in the UK, and she did postdoctoral training and also instructorship at the Rush ADRC. And she's interested in neurodegenerative disorders, cerebrovascular disease, aging, and cognition. But she also has been doing a lot of work in the digital pathology space, which she's going to talk about today. I'm really excited to hear about it because of all of the rich cohort that they and resources that they have at Rush. It's just truly amazing. And I also just want to give a quick PSA. If anyone has any topics that they would like to talk about in the future, feel free to send me an email. I would love to have to line up more speakers coming this fall. I'm doing a pretty good job of filling the agenda. I would like to have this monthly, but if anyone really wants to talk about their research or is dying to, please go ahead and send me an email at kurt.farrell@mssm.edu or send me a message during this call in the chat. So at the end of the talk, we're going to use the Q&A feature in Zoom. It's under the More tab if you can't see it, for questions. And with that, I would like to get it started, and I'll pop my email in the chat too if anyone wants to send me a line to present in the future. All right, take it away.
Thank you. Hi, everyone. Thank you, Kurt, for the very kind invitation for having me speak today. So I'm Alifiya Kapasi. I'm an assistant professor at the Rush ADRC, the Neuropathology Core co-lead for our Rush ADRC P30 grant. And today I'm going to talk about a publication that we submitted at JNEN, and it was published there last year. And it's on high-throughput digital quantification of Alzheimer's disease and associated infrastructure in large autopsy studies. So I know Kurt gave me a really nice introduction, but I thought I would take this opportunity to introduce myself and my background. So I have a PhD in developmental neuroscience. This was in Sheffield, England. It's where I'm born and raised. And my PhD was really focused on understanding mechanisms associated with neuronal migration. And I was using the zebrafish model. In some of my earlier work, I was using Drosophila model. But I really wanted to take my research into more human-based studies. And so I switched career focus to neuropathology. I joined the Rush team and spent a lot of time learning and studying diagnostic neuropathology for Alzheimer's disease and related disorders. And it was during my time here that I've really been introduced into this multidisciplinary approach for translational neuropathologic studies. And then in 2022, I was fortunate to receive a KO1 Research Scientist Award. And for these KO1s, part of the application is focused on training, and the other half of the application is focused on research. And so my training was really around this aspect of digital pathology, MRI approaches, biostats, disparities in ADRD, and clinical assessment. And my research program is really trying to understand vascular changes in the brain and the changes in the vasculature and how that has a role with Alzheimer's disease and neurodegeneration. So we know that the two main pathologic hallmarks of Alzheimer's disease are the extracellular accumulation of beta amyloid plaques and the accumulation of neurofibrillary tangles composed of misfolded tau protein. And we can see that these two pathologies are depicted here with the 4G8 amyloid stain and the 88 tau stain. And over the last several decades, there have been some really important revisions in the neuropathologic criteria for Alzheimer's disease. The initial criteria included CRAD, which is the consortium to establish a registry for Alzheimer's disease, and it really largely depended on the number of neuritic plaques in the tissue to give a likelihood of Alzheimer's disease pathology contributing to that dementia, and it would give a possible, probable, or definite likelihood. A few years later, the CRAD criteria was combined with the Braak staging criteria, which we know is the progression of neurofibrillary tangles in the aging brain. And then a few years later, there was a shift in sense of we can diagnose Alzheimer's disease pathology, and there doesn't have to be a dementia diagnosis linked. And so this criteria kind of separated the clinical diagnosis from the pathologic diagnosis, and really combines the CRAD, Braak staging, and Thal phase, which is the progression of amyloid plaques. But in all of these criteria rely on the highest area count or the topographical staging of pathologies. So this comes to the question of what are we missing? So many research studies have used stereology to go one step further for getting quantitative pathology data. Here I've just listed a couple of papers, but there are many more publications in PubMed that have used stereology in the context of Alzheimer's disease and in the context of postmortem brain tissue. But again, these methodologies do have its limitations as well. Generally, they are manual counting methods, and so they are open for inter-rater variability. Often there is a lot of effort required to really sit down at the stereology system and count these cells or count the pathologies you're interested in. And we are sampling a small area of interest. And so now we come to this new era of digital and computational pathology. And while there are some advantages of these approaches, there are still some limitations as well, which I will get to later on. So some of the advantages are if we have digital slides, we can prevent loss or breakage or fading of actual pathology slides. We're able to conserve valuable tissue resources. With many image analysis platforms now, we are able to deeper phenotype these brain tissue specimens and characterize the different types of patterns that we might be seeing in the tissue. And if there's a centralized digital slide repository, virtual viewing and ease of sharing becomes much more feasible. I want to highlight some studies that have actively leaned into these digital and computational approaches for morphometric analysis of postmortem brain tissue. So being able to utilize deep or machine learning algorithms, several groups have looked at different amyloid pathologies, neurofibrillary tangles, tau pathologies in other tauopathies, and also the vascular changes that we might be seeing associated with aging. And so at the RADC, we're really interested in community-based cohorts to study aging and dementia. We have 5 different cohorts for which some require brain donation and for some brain donation is optional. And so the religious order study and the memory and aging project, these 2 cohorts really started in the mid-1990s. And the other cohorts, and so for the ROS and MAP cohorts, brain donation is required. For the other cohorts highlighted in green here, these are the minority cohorts and brain donation is optional. So we have the Minority Aging Research Study, MARS, African American Clinical Core, and the Latino Core, which is the most recent core that has been developed. But essentially in all of our cohorts, the foundation of study design is essentially the same. We have tremendous participant outreach and building of relationships within the community. Once the participant enrolls in the study, we have an annual neurologic and cognitive evaluation. And then participants sign an anatomic gift act, and then we perform the brain donation and postmortem procedures. We do a neuropathologic diagnosis using the standardized criteria and different aspects of data collection. So one of the data collections that we have been historically performing at the RADC is quantitative data collection for amyloid and for tangles. We sample different brain regions that we're interested in. We prepare 20 micron pathology slides, And then we assess those slides by image J or stereology approaches. These slides are stained with amyloid or for 4G8 or for 88 for tangles. And our amyloid stain actually included a nickel enhanced staining protocol. So when there was positivity on the slide, that image would be a purple to black appearance on a no counterstain background. So really it looks as almost a binary image. And we would upload these into image J to get 8% positivity. And then for tangles, we were using the 88 stain with a counterstain, and we were manually counting cells, counting tangles across specific ROIs. And as I mentioned before, there were some limitations with this approach. It is labor intensive. One pathology slide takes about 45 minutes to 60 minutes to evaluate. And we're not evaluating the entire cortex. We're sampling about 5% of cortical tissue. So the goal of this study was to utilize digital pathology approaches to aid high throughput quantification of Alzheimer's disease pathology. And our goal too was to harmonize newly generated digital data with historical data. And I think for many of us, this is a really important fact because we are involved in these longitudinal epidemiologic cohort studies. And so if we change approaches or we change methodologies, we want to ensure that we're harmonized with what we plan to do prospectively and what we have done retrospectively. So at the RADC, we have the Aperio AT2 digital scanner. It's a very popular scanner that many other ADRCs and neuropathology cores have. There are other options out there as well, though. We like the scanner. I think it's a really nice system, very user-friendly. It scans about 400 slides, about 50 slides per hour. I don't think we've ever reached the capacity of 400 slides. And really some of this work was feasible because we had a supplement grant for our ADRC neuropathology core. And the supplement grant, the goals were to create this digital slide repository and implement these digital pathology workflows. So our digital pathology workflow is as presented here. We sample different brain regions that we're interested in. We stain those brain regions. So for this study, it was for 4G8 and 88 for amyloid and tau. We then scan the pathology slides, creating a digital slide. One nice feature of our workflow is that we have this automated data entry. And what that means is that we have an optical character recognition software, recognizing the letters and the characters from the slide label, and then auto-populating that information from the slide label to our internal digital slide hub. This is a really nice feature because it really has eliminated manual data entry and then also eliminates any errors prone to manual data entry. We can then virtually view these slides. Again, this is a really nice feature, being able to view slides on computer systems as opposed to being static at a microscope station. And then we have immunohistochemistry images, the automated quantification, the mask, the digital data mask, and then the analysis of digital data. So to develop any workflow and to get any workflow up off the ground running, it really does require a multidisciplinary team. And here we try to depict the different teams coming together to really show that this is a circular relationship. We have digital pathology and data management teams creating these workflow management systems. We have the data management team and statistics communicating about data flow. And then we have the digital pathology and statistics really optimizing and validating the data. And so I want to take a minute here to really thank all these individuals in the neuropathology lab, the biostats lab, and the data management team. It's really been a great learning opportunity for me, myself, and it's been a really nice journey to get this up off the ground. So for our beta amyloid pathology, we used a positive pixel algorithm. In general, these algorithms are pretty robust. They are available on ImageJ as well and other image analysis platforms. And essentially, it's using an intensity to detect a pixel. So when you have a large intensity, that means the pixel is brighter. So that's why the intensity will range from 0 to 255. And then it allows you to tweak your parameters to define what is weak, what is medium, and what is strong intensity. So here we can see that the 4GA immunohistochemistry is highlighting the amyloid plaques. And then we have the digital mask, which is really depicting the different intensity thresholds of that DAB staining. We wanted to validate these algorithms that we have built. And so we selected cases that already had historical amyloid data workup. And we selected these cases based on the different quartiles. And we specifically selected cases that had 0 amyloid pathology as well, that were historically counted as 0. And we evaluated different brain regions, and we had 413 slides in the data set, which corresponded across 93 participants. In the RADC cohorts, we have generally an older age at death. It was about 90 years. But you can see that the data set ranges across the Thal score. So we have 11% of participants with a Thal score of 0, and then there are different ranges across those different Thal phases. When we looked at the relationship between what our digital algorithm was outputting and what our historical data was measuring, we see a really nice correlation. One thing I'd highlight here is that even in the cases that were historically evaluated at 0, there is some minimal digital data output. And when we looked back at these slides, it really was that the digital data was capturing actual pathology. And so this goes down to really sampling. When you have low burden cases, low burden in pathology, it really is a sampling issue. We're sectioning the next level, and so maybe we're getting some pathology there. In addition, we also saw some parenchymal CAA in these cases that were historically evaluated as 0. For our tau tangles, we used a different algorithm. Instead of the positive pixel algorithm, we used a nuclear algorithm. So the nuclear algorithm really detects the size, shape, and intensity of these nuclei. If a nuclei is recognized, but there is no DAB staining, it will color code it as a blue. If there is some light staining, it will be yellow, and the darker the staining, the different thresholds, it will color code it as orange and red. And so you can see here that the 88 immunohistochemistry, you can see the neurofibrillary tangles, there's some tau positive threads in the background, and then the digital mask is really highlighting these tangles. Again, for our data set, we wanted to validate this algorithm. So in a very similar way to what we did with amyloid, we selected cases that had historical tangle data workup across the different quartiles. We specifically selected cases that were historically evaluated as 0. And then we worked up these different brain regions. Again, our mean age at death is about 90, and we have a range of Braak score. In general, because we do have a older age of death in our data set, we have very few individuals or a limited number of participants that actually have a Braak score 0, 1, or 2. But you can see that there is a much higher representative of the data set in the Braaks 3 to 6. Again, when we looked at the correlation between what the digital algorithm was outputting and our historical measure of tangles, we see a very nice correlation. And so this is really a really nice way of validating that these approaches are correlated to what we are considering the standard, right? The stereology is our standard data. The image analysis was our standard data for amyloid. We then wanted to look at some region-to-region correlations. And so for both amyloid and for tangles, so the top half of the slide is for amyloid, the bottom half is for tangles. And we see that the region-to-region correlations are pretty high for amyloid. This is what we expect. And then as we look through the different Thal phasing, the burden of amyloid is increasing. Again, very established patterns. This is what we expect. And this analysis was actually done on a larger data set. So we had a validation set, and then we ran these algorithms on a larger data set, so 341 participants for amyloid, 197 participants for tangles. For the region-to-region correlations for tangles, there is a little bit more variation. Again, this is what we expect. We see that the hippocampus and entorhinal are highly correlated with each other. Midfrontal and superior frontal are highly correlated with each other. And calcarine and hippocampus are less correlated. Again, this is an expected association that we would see. When we look across Braak score, so 0 to 6, we see that in the earlier Braak stages of 1 and 2, it's really the involvement of the entorhinal cortex and hippocampus. Braak score 3, we start to see inferior temporal tangles being more involved. Braak score 4 and 5, the neocortical regions, and then Braak score 6, we see the calcarine cortex being involved. We then also examined correlations across clinical and neuropathologic diagnoses. And again, we see very established patterns that we would expect to see across clinical diagnosis and pathologic diagnosis of AD. So then part of our second goal was to harmonize this new digital data with the historical data. So we used these statistical approaches where we ran some linear regression models. So for each validation data set, we ran a linear regression model where the historical data was the predictor and the digital data was the outcome. And so from each of these linear regression models, you can utilize the coefficient term. And that's what we use to transform the historical data to a predicted digital score. So when the historical data was 0, the digital score would map onto 0. But if there was positive pathology in the historical data, then it would predict what the digital score would have performed. So we created these new computated variables. And if there was raw digital scores available, it would use that. And if there was historical data, it would transform it and use the predicted digital score. So then we wanted to really assess robustness, right? We have these kind of 3 different data sets, and we want to ensure that they're acting in the same manner. And so we ran a principal component analysis. We have a digital data set of digital amyloid. We have a historical data set of the historical data, and then we have the harmonized data set. And we can see that the principal component analysis really reveals this 1-factor loading. Which makes sense, right? We saw very high regional correlations between all those 8 brain regions. So this was very reassuring to see that this pattern was existing in all of these different data sets. For tau tangles, the PCA really revealed 2 components. Again, this is, I think, something we expect. The neocortical regions kind of hanging very closely together, the entorhinal hippocampus regions kind of hanging together, and then this inferior temporal region really was loading onto both factors, factor 1 and factor 2. And this relationship was seen in all of these 3 different data sets. So when we looked at the digital data for tangles, when we looked at the historical data for tangles, and when we looked across this harmonized data set. So I mentioned that there were some limitations with computational approaches. And I think one of the big limitations is that really all downstream analysis relies heavily on having high quality digital slides. So we try to implement multiple QA/QC checkpoints within our workflow to really ensure that we do have high quality digital slides. We evaluate for staining or any slide preparation artifacts, as well as scanning artifacts. This is done routinely on a weekly basis. When we outline for our interests, we also examine for tissue-related artifacts that could be impacting any downstream analyses, as well as when we have new staff members joining or new raters, we perform inter-rater methods to really ensure that the ROI outline is similar across different raters. And then we also perform a digital data review. And so we have these monthly QC data box plots, which is a really nice tool because we can really start to understand the data structure and then query some of the upper tail of the data structure. And then we also have some tracking reports to ensure that the workflow is progressing. And so just as an example of the QC data box plots, so here you can see there's a box plots for amyloid and for tau by each region. And you can see that the upper tail, we can kind of select these dots and see which slides they are, and then go into the system and verify that this is what we expect. So moving on from Alzheimer's disease pathology, I wanted to highlight some of our more recent work in the space of digital pathology. We're interested in other pathologic markers associated with AD, associated with neurodegenerative diseases, and one of them is microglia. So we're trying to leverage the Halo image analysis software. So Halo is another platform developed by Indica Labs. And we're utilizing their machine learning classifiers to first, can we recognize these microglial cells? And then secondly, can we characterize these microglial cells as being less reactive? So kind of more stage 1 versus more reactive, being more of a stage 2, based on morphology. So the box highlighted here is a cell that we would kind of call as more reactive. It's more larger in cell body sizes, has a little bit more thickened processes, and some of the other cells here highlighted in red are a little bit more smaller in cell body size. And so we've performed some kind of preliminary checks to see how this Halo digital output for microglia density is working with our stereology microglia density that we performed counting the cells. And we see a very nice correlation between both approaches. So very reassuring that the digital output is related to what we would consider as the standard. And so we're moving forward with this protocol and this approach in different settings. And then another glial marker that we're interested in is astrocytes. So we're interested in gliosis or astrocytosis. This is work led by Sonal Agarwal in the lab, and she's developing an algorithm to detect GFAP, which highlights the astrocyte cells. And so with that, I'd just like to give thank you to the Rush Alzheimer's Disease Center. It's a really great team to work with, our neuropathology lab and staff members, especially Ryan Johnson and Shivani Mandel, and then the participant and brain donors of all our cohorts and our funding bodies. And if you're interested to see what available data we have for sharing, then please go to the link and see our RADC resource hub.
Thank you so much. That was an amazing talk. I loved seeing how you were correlating the new digital techniques with some of your historical data. I had one quick question to kind of get it started. I'm curious about, first of all, how many different regions of interest would you say you guys have annotated thus so far? And when you are annotating them, how are you keeping track of how you do it? Is there online manuals? And if you're teaching someone who doesn't have a background in neuropathology, how do you make sure that they're annotating the right region? Do you just use like features off the slide, or just what goes into that?
Yeah, so it's very timely. We actually recently had a staff member join who has limited experience with annotating regions. And the approach that we take is that we try to do these inter-rater reliability checks. And so we're evaluating multiple different regions. Most of the regions are cortical. So we really are able to distinguish the gray matter from the white matter. And so those generally tend to be very reliable when you have different raters rating them. I think some of the more variable regions can be the amygdala, the hippocampal subregions, perhaps even entorhinal cortex. And so that's where we rely on some of our inter-rater methods, where our stats teams helps us kind of identify how reliable these different raters can be in these approaches. But yeah, no, I think it's a challenge. And it really depends center to center. I'm pretty sure what we are doing here might not translate over to what another center is doing, even for those same regions, right? So I think that's where some of the standardization discussions would be really important to have.
Yeah, absolutely. We found on our end, we've had different versions of kind of the system that we're using to annotate. Version 1 was, very crude, at least in the hippocampus. We just circled the hippocampus proper. And then as we started to get more advanced and asking different questions, we created version 2 and locked it down, where we tried to get subfields. And now we're trying to do even more subfields. So it keeps getting more complicated and complicated. To the crowd, if anyone has any questions, please feel free to put it into the Q&A. I had one other question that I was curious about. How has your method of quantification worked outside of kind of the standard stains, like the AT8? I mean, I know you're doing AT8, and what if like a researcher comes up to you and says, I want to look at STX6 or something different, something that doesn't have, is the Aperio good at picking up on those patterns?
So I will say that the pipelines that we have in place right now, so for amyloid and for tangles, we're using the Aperio system, the ImageScope image analysis platforms. We have yet to translate that onto different cohorts or even different stains. So we're really utilizing what we are doing in-house. And I think that that's an important point. I think, that is something that we should be thinking about and trying to translate these things across centers, across sites. They become much more robust that way. I will say that I feel like just working both with Halo and working both with ImageScope, there are some limitations with what you can do with ImageScope as opposed to what you can do with Halo. I feel like there is a little bit more flexibility when you have these machine learning classifiers or some deep learning embedded into your algorithm. And as you know, I mean, you're definitely involved in the computational pathology approaches. So I think the more complex and heterogeneous a pathology is, I think it requires more sophisticated tools to be able to capture that.
Absolutely. Absolutely. That makes sense. We have a Q&A question here from Larry C. Walker. It says, can you elaborate on distinguishing CAA from parenchymal A beta plaques? Can the algorithm reliably distinguish them, including capillary CAA?
Yeah, that's a great question. And so we have kind of two arms of our data collection, right? So we have our neuropathologic diagnoses. We're making a pathologic diagnosis of Alzheimer's disease, and we are making a kind of pathologic diagnosis of CAA, and those are very separate. For our data quantification, our algorithm for amyloid pathology does incorporate presence of parenchymal CAA. We are in the midst of trying to utilize the Halo system, whether we can really differentiate plaques from CAA. It seems like there is some promise there. But currently, our amyloid pathology algorithm does incorporate the parenchymal CAA. And that was what we were historically doing as well. So we're really trying to leverage applications that have been in our lab for many years and trying to replicate that so that we're not changing our data structure too much.
Awesome. Thank you so much. I was wondering too, moving forward, I know some of the people on the call have systems where you can view their slides online or even share and down them digitally. Harry Hartunian has his Charcot platform. Has Rush thought about making some online viewers and not necessarily sharing, but at least showing the way that you're annotating these regions? Because I think moving forward, we're all trying to get to a place where we can share slides just like we've shared genetic data in the past, even though they're quite large and there are some hurdles to doing so. But I was wondering if Rush had thought that they would be moving in that direction anytime soon.
Yeah, I think that's a great question. It's a really great point. Something I've thought about personally. And yeah, I think it'd be great if people want to collaborate and with us to really facilitate that. Right now we share slides, but we don't really have a kind of open access virtual viewing system. And I think, yeah, that'd be really a great tool to have. Absolutely.
Yeah, there's definitely some barriers in getting those up and being able to host it. Um, all right, so with that, I see a hand waved by Dr. Duggar.
Yeah, I just wanted to congratulate you first and foremost on this study, Dr. Kapasi. And you touched upon it a little bit through the talk, but maybe just for some of those people on the line, maybe emphasize the resources again. You emphasize just the cross-disciplinary dialogue, and then an administrative supplement that got this off the ground. But anything else you want to share for some of the people on the line that are interested in doing these types of studies, kind of like a lessons learned?
Yeah, I mean, definitely it's all computational, right? So we do have 3 servers that is really running this workflow. A server that has the digital slides and the metadata attached, a server to actually run the analyses, and then another server to kind of handle the analytic output. So those are kind of the hardware, I guess, the hardware of the system to really facilitate this workflow. And then we also have, at least for now, a lot of storage capacity. I think we have about 25 terabytes of storage capacity for these slides. Of course, that's going to differ whether you scan in 20, magnification of 20 or magnification of 40, of how fast you take up that storage capacity. So those are some of the, I guess, other aspects of the workflow I did not mention, in addition to the expertise and personnel across the different disciplines that help, advise you and help you kind of build this out.
And then with that, you talked about the supplement. What other sort of resources, like institutional, that you got to try to do this? Because I'm hearing the servers and this and that, and how much is that supported by grants versus philanthropy versus takes a village, right?
Yeah, no, absolutely. I mean, I think we were fortunate to have our scanner, which was more of an institutional, and the RADC, the ADRC supplement grant really funded the development of the workflow. But yeah, I mean, I think we were fortunate to have institutional support on really building out some of these other pieces of it.
Amazing. Well, I don't see any more questions in the chat. So I think with that, we'll conclude today's talk. Again, can't thank you enough. I can't thank everyone enough for joining. Please feel free to email me if you want to speak at one of the upcoming month's chat. And I look forward to seeing everyone again in about a month. So have a great rest of your afternoon, everyone. Thank you.
Good. Thanks, everyone.